So, the key takeaways from our three presentations at AACR, our first response rate, we saw deep responses, MRD negative CRs in 4 out of the 7 patients. This MRD negativity means no detectable AML for the best available test away at each local site. Um, we also have correlative data, which shows that our logic gate is doing what it's supposed to do in terms of targeting AML and the leukemia stem cells and preserving the healthy cells. And we had preclinical data, which further parses out the contribution of the NOT logic gate. So overall, a terrific AACR for us, and a lot of confidence in our approach, not only to move to forward for AML, but also apply these logic gates for other cancers with high unmet need like solid tumors.
Welcome back, and thank you for joining us for another Virtual Investor What This Means segment. My name is Janine Thomas. I am CEO of JTCIR, and I will be the moderator for today's segment. Today, we are featuring Senti Biosciences. Senti Bio's lead clinical program, Senti 202, demonstrated positive preliminary clinical results in the treatment of patients with relapsed refractory AML. So today, we're going to break down what this means for Senti and potentially for the AML community. I am very pleased to be joined by Dr. Kanya Rajangam. She is President, Head of Research and Development, and Chief Medical Officer of Senti Bio. So before we get started, I just want to remind our audience that Senti Bio is publicly listed on NASDAQ and trades under the ticker SNTI. And during today's discussion, the company will be making forward-looking statements, and I encourage you to view the company's website at sentibio.com for the latest filings and information. So we're going to dive right in. So Kanya, you had an exciting AACR conference with new data presented. So congratulations on that. And you achieved an overall response rate of 71%. So that means 5 of 7 evaluable patients with deep, durable responses in your phase 1 study. Can you explain what these results mean for Senti 202's potential as a first-in-class therapy for relapsed refractory AML?
Thank you, Janine, and thank you for having us. We are super excited about our AACR results. As you said, we have 5 out of 7 patients saw some blast reduction with for that ORR. That included 4 patients who achieved a CR, a composite CR, forgive me, including 3 who achieved a CR with full hematologic recovery and 1 CR with partial hematologic recovery, which meant that their normal counts came back, but not all the way up to a CR. So all of these CR/CRh patients were also MRD negative. What that means is that by the most sensitive method available at each site's local per their local standard of care, there was no residual AML disease detected. So that's really exciting. And we know that MRD negative CR translates to durability, and that's indeed what we are seeing as well. Just stepping back for a minute, late last year, we presented data from our first cohort, first dose level, and we saw 2 patients achieve MRD negative CR out of 3. And at that time, our follow up was about 3 months or so because that's how long the patients had been on trial. Fast forward now to AACR, those patients continue to maintain their remission, showing that indeed that MRD negative CR, as we had, predicted and as actually data from the field shows, does translate to that durability. So now our durability, we have our 4 patients, 8+ months out, 7+, 5+, and 4+, and all of them are maintaining their remissions. The other thing that comes out with this early piece of data is how well tolerated Senti 202 is, which means that our how we are developing Senti 202 is for potential outpatient use. So assuming the safety continues, that means we should be able to give this in a setting where you don't have to be hospitalized just to get the cell infusion, potentially increasing access for patients.
Okay, very exciting and very encouraging, Kanya. So, Senti 202 uses a logic gated system to selectively kill cancer cells while sparing healthy ones. Based on the MRD negative results and bone marrow cytoph findings, how confident are you that the logic gate approach is working as designed in humans?
Thank you for that question. So what we so one of the things that's really exciting for us about our AACR data reveal is not only the clinical data, which we just talked about, the response rates, the durability, showing in patients that the leukemia stem cells decrease after they've received Senti 202. In addition to that, what we also presented is preclinical data where you're able to isolate the different components of our logic gate circuit to see what contributed to which clinical phenomena, so to speak. And there, in our humanized mouse model study, which was actually presented in a poster today in Chicago, what we show is that with the having that NOT gate targeting endomucin is necessary to see that protection of the hematopoietic stem cells and the endomucin expressing hematopoietic stem cells. So, putting all of this data together, the humanized mouse model, the correlative, and of course, our clinical data, this gives us a lot of confidence in our approach. And in fact, in our pipeline, we have early discovery efforts to expand this into other high unmet need solid tumor cancers.
Thanks for that. So you identified a preliminary recommended phase 2 dose without any dose limiting toxicities. What does this tolerability profile suggest about Senti 202's potential for combination therapies or broader use beyond AML?
Thank you. So that's one of the things that, going into this, we know that CAR NKs and NK cell therapies in general are better tolerated than T cells just because their inherent biology is different. They have some modest expansion. They don't have this huge outpouring of cytokines in general. So seeing that similar safety profile with Senti 202, continues to support what we are trying to do here, which is eventually develop this for outpatient use, which could make a big difference in terms of patient access. So you don't have to come in and be hospitalized in order to get this therapy. We are also thinking, because of the tolerability profile, one of the other things, AML, as with most cancers, ultimately is a way in which you combine different therapies to really maximize the benefit to patients. So, having such a clean profile, as you mentioned, certainly lends itself to doing that more readily. So, for instance, if we were go to newly diagnosed patients, as an example, it gives us more flexibility in what we can do because we are not concerned about additive toxicity.
Many patients achieving CCR moved on to transplant. How should we interpret the role of Senti 202 as a bridge to transplant, a standalone therapy, or potentially both, depending on patient characteristics?
We are looking at it as both. In our phase one trial, patients come on, and those who achieve a CR, and this is true whether they're in a phase one trial or in any other trial, or even with current standard of care. Once you achieve a CR, the current standard would be to consolidate with the transplant in patients who are able to tolerate the transplant, and that's what we are seeing in our trial. Not all patients can get a transplant either because of some other condition going on or they don't have access to a donor. And so, in our trial, which is what has happened as well, we will be able to see this reflect what happens in AML treatment outside of a trial. Some get transplant and some don't. And so we will be able to get that data across the board. And that's how we're thinking of Senti 202 as a standalone therapy. And then those who can go on and get consolidated with transplant, that's good for them. But those who cannot, we are hopeful that this early durability, that MRD negative CR can translate to meaningful benefit for patients even without getting a transplant.
So, Kanya, now that you've shown early signs of efficacy and safety, what are the next steps for the Senti 202 program? And what should we expect in terms of future data or study expansion?
Yes, so we have declared preliminary recommended phase 2 dose or RP2D. So, what happens next is that we confirm it, which means we put on a few more patients at that dose, look at the totality of data, safety, efficacy, pharmacodynamics, PK. Once we've confirmed it, what happens next is additional expansion cohorts where we really focus on one tumor type, either AML, and we also have the ability to bring on other tumor types, MDS, and so on. But for this year, we are laser focused on AML. So that's what we would do is confirm the dose, expansion cohort, so that we can get more patient numbers, more durability data, and more response data. And overall, we are very excited by what we are seeing, what we saw in December, what we presented today at AACR, and we are continuing the study. So we hopefully continue to see the same types of data for us to then proceed to next steps, which is to start thinking about larger registration supporting trials.
Appreciate that. So very exciting times here for your Senti 202 program and for Senti Bio as a whole. And with that, this concludes our Virtual Investor What This Means segment featuring Senti Bio. As a reminder, Senti trades on NASDAQ under the ticker SNTI. And if you like what you saw today, I encourage you to visit sentibio.com for more information on the company and to sign up to follow the company to receive their alerts, as well as follow their social channels on X and LinkedIn to stay current on the latest information. We'd like to thank everyone for joining and wish you a great rest of your day.